Hello students, welcome to chapter 6. We're going to be talking about energy in biological systems. This means through enzymes, through Gibbs free energy, through equilibrium, thermodynamics, everything like that. Okay. Pretty much energy is defined as the capacity to do work. It exists in many different forms through heat, chemical energy, X-ray energy, gamma rays, all these different types. So it can be converted from one form to another. So in plants, you can get radiant energy from the sun, and that can be converted into chemical energy through organic molecules. You can convert kinetic energy into electrical energy. So you can do that through many different forms, like water systems, through turbines, or sonic energy, these ionizing energies or whatever. All forms of energy can exist in one of two states. We have kinetic energy, so that's movement, and then potential energy, that's stored energy. Usually that's still energy. So examples of kinetic energy is thermal energy, that's in heat, so that's moving particles. Mechanical energy, this is energy in motion. Electrical energy, this is particles, usually electrons moving. Potential energy examples, chemical energy, this is actual molecules or chemicals stored in those, energy stored in those molecules. Elastic energy, if you stretch like rubber, that would be elastic energy. Nuclear energy, this is like ionized energy. So if you have if you have radioactive isomers, they have nuclear type energy. Thermodynamics, if we have the study of energy and its transformations, that's the study of thermodynamics. Whatever the scientist refers to in that system, right? Let's say it's an ecological system, right? We have a boundary within that system. Everything outside the system is its surroundings. So you have whatever boundary and its system, right? So our digestive system is a type of thermodynamic system. And then you have three different types of systems. You have an open system, a closed system, and an isolated system. Okay? The open system allows heat and matter to be transported out of that system, in and out of that system. Closed system only allows for heat to be transported out of that system. Matter stays within the system. Okay? An isolated system is completely closed in. It doesn't allow for any matter or heat to be transferred out. So if you think about it this way, an open system is completely open. It's open to all its surroundings whatsoever. So all living organisms like you and me are open systems, right? We can input food, we can output waste. Heat and matter go in and out of us. A closed system, let's say that we have some type of like bag, and then it's clear. right? It allows for heat to be transported out, right? But no matter is coming out. But let's say we have a bag that is not clear. It's painted over with spray paint. That's an isolated system. It doesn't allow heat to come out, or like it's a box. It doesn't allow heat to come out. It doesn't allow anything to come out, or matter or anything. That's an isolated system. So the first law of thermodynamics, energy can be transferred from one form to another, or from one place to another, but it cannot be made or destroyed. Right? Energy itself can't be made or destroyed in our context of biology. So in the principle of conservation of energy, this is what it's defined as. It can't be created or destroyed because it's conserving that energy. It can be transformed, but it can't just blip out of existence or just blip into existence. So any process that involves energy change, the total amount of energy in a system and its surrounding remains constant. So we can take this overall equation here. You have the total energy equals whatever heat was added to the equation minus the work done. Right? You have to have some amount of work done, and then there has to be some amount of heat that's been erased from the equation. Right? Because there's always going to be some amount of heat whenever there's an energy transfer. So the second law of thermodynamics, the total disorder meaning some type of chaos, that's entropy. All right, so the total entropy of a system and its surroundings of a system and its surroundings. So if you have the system that includes the surroundings, it always increases, although the total energy does not change. All right, that would violate the first law of thermodynamics. All right, so the total entropy always increases, but the total energy doesn't change. Living organisms seem to decrease in entropy as they grow, but when nutrients and waste products are considered, total energy remains constant and entropy increases. So it seems to decrease in entropy, but when you consider all the nutrients and waste products that are put into your system and putting out of your system, the total of all of that, entropy actually increases as you grow. So you yourself, in your context, seem to decrease in entropy, but in your surroundings plus your entropy is increasing, but still your energy remains constant. Free energy and spontaneous reactions. So a spontaneous reaction is a chemical or physical reaction that will occur without any input of energy. So reactions tend to be spontaneous if the product has less potential energy than the reactants. So it has to be more random than the reactants. So if you have the products over here and the reactants over here, if it has less potential energy over here than the reactants, it's going to be more spontaneous. The potential energy in a system is its enthalpy. Okay, that's just another word for it. Reactions that release energy are exothermic, right? So if they're releasing energy, it's gonna feel warm. So it's exothermic, it's releasing energy. Exo meaning exiting heat. Reactions that are absorbing energy, which means it's bringing in heat, it's bringing in energy, are endothermic. Endo meaning in heat. It's bringing in heat. It's going to feel cold. Now we're going to talk about Gibbs free energy. So Gibbs free energy, this is an important subject. The portion of a system's energy that is available to do work is called free energy. Okay, so this is kind of like the free availability of memory on your computer. The change in free energy can be calculated for any chemical reaction in the formula. So change in G, change in Gibbs free energy, equals the change in enthalpy, or the change in potential energy, minus the absolute temperature in Kelvin, times the change in entropy. That's the change in randomness, or the change, not the change in randomness, the change in disorder. You have all this, and it's probably making no sense to you whatsoever, but we'll talk about it. So for a reaction to remain spontaneous, the change in Gibbs free energy must be negative. Okay? That means that the change in enthalpy has to be less than the change in degrees Kelvin times the change in entropy, because that would remain negative. In some processes, such as the combustion of methane, there's a large loss of potential energy. So negative enthalpy, or that change in enthalpy, dominates in making the reaction spontaneous. So it gives that Gibbs free energy the ability to remain negative. In other reactions, such as the melting of ice, a decrease in order, a decrease in that entropy, it dominates the equation, right? So that also gives it the ability to remain negative. So the spontaneous reaction doesn't exactly mean that it's just going to blow up. It just means that at a certain degree, it's going to give it a reaction that will change the context of whatever chemical that you're working with. So ice melting, that is a reaction. It's changing from a crystalline structure to a more liquid structure. Equilibrium point. This one's a little bit difficult. So a state of balance between the opposing factors push the reaction in either direction. Right? So you're going to have some balance between one reactant and the product. The reactants run in the direction of completion, either toward the reactant or the product, until they reach an equilibrium point. As a system moves towards equilibrium, its free energy becomes progressively lower, and it reaches towards that equilibrium point. Okay, and that equilibrium point is when free energy becomes zero. To move away from equilibrium requires free energy, and thus becoming not spontaneous, right? Because, like I said, in order for a reaction to become spontaneous, G has to be less than zero. So in order for the equilibrium point to remain at that point, it has to equalize to where G becomes zero. The more negative change in G is, the further towards completion the reaction will move before equilibrium is established. Okay, so you can see over here on the side, we have the reactant and the product, right? So at some point, we'll have the reactant moving towards the product until it reaches equilibrium. Once it reaches equilibrium, it stops, and G becomes zero. Many reactions have a change in Z that's near zero and are readily reversible by just adjusting the concentration of products and or reactions just very slightly. So if you add a little bit of product or add a little bit of reactant, you can reverse that reaction depending on what it is. So you can just have that double arrow right in the center, and then you can add a little bit of one side and more reactant will appear or more product will appear. So reactions in living organisms. So how does this fit into us, or how does this fit into animals or plants or whatever? Many reactions in living organisms, they don't reach equilibrium. Because we're open systems, if you were to reach equilibrium, most of the time, that's when they are dead. Okay? Because we have energy-rich molecules, we have enzymes, we have things that are working inside of us, that type of equilibrium doesn't exactly work. So we have a supply of reactants, we have a supply of products that are going on. So they don't accumulate. We always take in those products. So if you reached zero, you'd either stop eating or you'd stop taking in nutrients. And most likely that would be at a time when you're dead. Exergonic versus endergonic reactions. So endergonic reactions, these are reactants that must gain free energy from its surroundings. So the system must gain free energy from its surroundings in order to form the product, right? So the free energy is positive because the products contain more free energy than the reactants. So the reactant is not spontaneous, which means that it has to be more than zero. If it's trying to gain free energy, it's not spontaneous, it has to be more than zero. And then we have exergonic reactions. Reactions that release free energy. These are ones that are spontaneous. G is going to be negative because it's producing energy. It's containing less free energy than the reactants. right? So if it's spontaneous, it's going to be less than 0. It's going to be releasing that energy. Some other metabolic pathways. We have catabolic pathways. These are things that break down simple molecules or break down complex molecules into simpler compounds. Okay? Think about it like if you broke down starch, if you have a starch cluster, and if you broke that down into simpler glucose molecules. If you broke down protein into smaller amino acids. In order to do this, you have to use that energy, right? So overall, G is going to be negative. I'm sorry. In order to do that, energy is released, so G is going to be negative, right? This is a spontaneous reaction. Catabolic reactions are spontaneous. They can just easily break down. Now, anabolic pathways, you have to use energy to build. So if you have smaller molecules such as amino acids and glucose molecules and other things, and you build them up into larger molecules, you have to use that energy, right? So overall, G is going to be positive, so you have to use energy in order to make that. This is not spontaneous. If we have adenosine triphosphate, this is the major energy currency of the cell. The nucleotide adenosine triphosphate consists of 5 carbon sugar, right? So this is linked to the nitrogenous base, adenine, and a chain of 3 phosphate groups, right? Adenosine triphosphate. The negative charges of the phosphate groups repel each other strongly. This makes the bonding arrangement unstable. So if you have those bondings, it wants to try and pull itself apart. So if you try to remove that, it's a really spontaneous reaction that makes that free energy release once it tries to bind itself, or once it tries to break itself apart. So this hydrolysis of ATP releases about negative 7.3 kilocalories per mole. That's a lot of energy per phosphate group. And this is what powers almost every reaction in the cell. And this is what your mitochondria make all the time. In the process of energy coupling, when ATP is hydrolyzed, terminal phosphate group is transferred to reactant molecule, right? Involving an endergonic reaction. So, if you have the addition of a phosphate group to a molecule, that's called phosphorylation. Right? So if you add the phosphate to something, it's called phosphorylation. Approximately 10 million ATP molecules are hydrolyzed and resynthesized every second in a typical cell. So that's a lot. So what is energy coupling? Without ATP, reaction is not spontaneous because G is positive. So if you took this glutamic acid and ammonia and put it into glutamic or glutamine, that's not spontaneous because it's positive 3.4 kilocalories per mole. It's not a spontaneous reaction, right? But if you took ATP and hydrolyzed ADP and phosphate out of it, that's negative 7.3. That is a spontaneous reaction. So what happens here is the ATP binds a phosphate group to the glutamic acid to make it glutamyl phosphate, right? And then the glutamyl phosphate binds with the ammonia to make glutamine and kicks off the phosphate. Overall, the net G is negative 3.9 kilocalories per mole, thus making it spontaneous. So this complex type of reaction makes it spontaneous, although it does add an extra step. That's what energy coupling does. That's how it makes it spontaneous, even though it does kind of add an extra step. Another way to get through these reactions is by using enzymes. So a catalyst is a chemical reaction or chemical agent that accelerates the rate of reactions without having to be changed by the reaction. So the catalyst doesn't change or anything, it just activates the reaction and helps whatever biological molecule that it needs to help. The most common biological catalysts are enzymes, right? So we know what enzymes are. They're type of proteins that go through and bind to certain biological molecules and, do their work. Enzymes don't alter the change in Gibbs free energy, okay? Not like ATP does, but it does change the path that the reaction takes. So for example here, if we have the activation energy without the enzyme and the activation energy with the enzyme, there's a lot less energy being released by the reaction. So it doesn't take as much energy to do this reaction. Like, if you see the curve there's a lot less free energy needed to do the reaction than there is to do the reaction without the enzyme. And sometimes that makes the difference. Enzymatic reactions, the 3D structure of a protein determines its purpose. And this is its conformation. So in enzymatic reactions, an enzyme combines briefly with reacting molecules, and you can tell this by what it's called van der Waals forces. So it just binds briefly to it, and it just goes, and it opens and it closes, and that's all it does. Releases unchanged when the reaction is complete. The reaction that the enzyme acts on is called a substrate. So whatever molecule that it's reacting on is called a substrate. Each type of enzyme catalyzes the reaction of a single type of substrate molecule. So you have one enzyme, one substrate. So it has to remain at a specific 3D structure. Otherwise, it will not fit that substrate. And it's called enzyme specificity. If it doesn't have a specific 3D structure, it will not fit that enzyme and it will not do its job. The substrate interacts with a small pocket or groove, and that's called the active site, right? So if you have that substrate coming in, it'll just click with it and then release it. When the substrate binds at the active site, both enzyme and substrate molecules are distorted. This makes the chemical bond in substrate ready for reaction. It's called an induced fit. Once the enzyme substrate complex is formed, catalysis occurs and the substrate is converted into one or more products. And you can see that on the side here. So you have one type of substrate, it'll come in, do its job, and then release. Sometimes enzymes will need other factors, other things that help it do what it needs to do. And these are called cofactors and coenzymes. So cofactors are non-protein groups that bind to the enzyme for catalytic activity. Some of these are metallic ions such as iron, copper, magnesium, zinc, manganese, whatever. Right? So these are just little ions or particles that can bind to the protein in order for it to do its job. Other molecules we can say are coenzymes. These are larger molecules, often derived from vitamins. So these coenzymes bind loosely to the enzyme. They can bind tightly to the enzyme. It just depends on what type of enzyme it is. Other factors that can affect enzyme activity. So you have competitive inhibition, inhibiting factors such as things that can actively bind to the enzyme itself. You can actually take medications that can bind to a specific enzyme and bind to the active site. That can actually slow or just stop the reaction altogether. Non-competitive inhibition. There are other things that can bind to other sites other than the active site. So if you have the active site over here and it just binds over here, that can reduce the reaction of the active site. It can change the formation of the active site. Okay, so we can see here competitive inhibition that actually goes directly to the active site, and then non-competitive inhibition that goes in another place that can affect the way the active site is formed. Another thing that can affect enzyme activity is regulation. So certain enzymes will make certain things inside your body that need to be regulated. Okay, so things like lactose. If you have too much of lactose and you're not drinking things that have lactose in it, you don't need that much lactose in your system, right? So it can stop making that. Right, so it needs to be regulated. So in allosteric regulation, enzyme activity is controlled by the reversible binding of a regulatory molecule to the allosteric site. This means that it'll bind to a certain part of the enzyme, and it'll only open up if there's a need for that enzyme to work. So binding an allosteric activator induces the high affinity state and increases enzyme activity. On the other hand, an allosteric inhibitor induces the low affinity state and decreases the enzyme activity. So if the allosteric inhibitor comes through, it'll close that active site, right? And the substrate won't bind to the active site. Another type of regulation is called feedback inhibition. So if you have an excessive accumulation of a product, that product will go back and inhibit the enzyme reaction. So it feeds back to the beginning of whatever that enzyme reaction is, right, and stop it from happening. So let's say that you have threonine, and then the enzyme 1 makes intermediate A, enzyme 2 makes intermediate B, enzyme 3 makes intermediate C, enzyme 4 makes D, enzyme 5 makes isoleucine. Well, if you have too much isoleucine goes back and inhibits enzyme 1. And then all the intermediates stop happening. There are other types of enzymes that we call ribozymes. And in the earliest forms of life, in the RNA world, as we call them, these ribozymes were a group of RNA molecules that accelerated the rate of certain biological reactions without being changed by the reaction. So these were types of enzymes in like those earliest forms of life. So neither DNA nor proteins played critical roles during these times. Some ribozymes catalyzed the cutting and splicing reactions that remove surplus segments of RNA molecules. Ribozymes in ribosomes catalyze the linkage of amino acids and protein synthesis. They acted as both enzymes and informational molecules. So when cellular life first appeared, they think that these acted as kind of ribosomal type things. So whenever they needed to be used for genetic references, these are what made the proteins. Other inhibitors of enzymes, many enzymes are regulated by chemical linkage in ions, so functional groups, other molecules, things like that. So in order to break these, you have to use specific conditions, right? So an enzyme dependence on pH is typically due to ionization in the amino acids. So if you change the optimal pH, you can denature that enzyme. And of course, temperature affects all those chemical reactions. So temperature can drastically affect the way that the protein is formed. So they can break those hydrogen bonds, it can break all those other bonds within the protein, it can denature those. Okay, for your attendance assignment, what I'd like you to do is try to find four ways that you can denature enzymes. Okay, make sure that you can get those into me before tomorrow, Friday. Okay, and then I think we're good for that. I'll go over the lab review and then make sure you go over that. All right, I'll see you later.